Okay. In the past, with a lot of the questions that I was seeing for the higher level papers, a lot of the questions they used for questions like 1 through 20 were similar to or the same as what was on the standard level sections. But what they're actually doing now, or what I've noticed in some questions more recently, is that even though these questions are technically talking about things in the standard level, the SL or the core portion, for this, in order to be able to answer this question correctly, you need to know some of the higher level enzyme interactions like competitive inhibition, end product inhibition, allostery as well too. So even more evidence that suggests that in these final days before the exam, you should be really making sure you see the connections between different parts of the syllabus. I cannot emphasize that more. So here's this question. It says, what happens when the substrate concentration is increased in an enzyme catalyzed reaction? Well, from your basic knowledge, you should know that enzyme reactions can be affected by three things. You've got temperature, you've got pH, and then you've got substrate concentration. And this one, substrate concentration, well, I know that if I increase the substrate concentration, the enzyme catalyzed reaction also increases, but it starts to slow down. And the explanation for that is because the active sites are being filled up. The more and more substrate there is, the less enzymes and active sites there are available to actually break down the substrate into their products. So that's my prediction here when I read this. What happens when the substrate concentration is increased in an enzyme catalyzed reaction? The rate of reaction is actually going to be decreasing to a steady point. But then when I look here, I see none of those are the actual answers. So I really need to use my basic knowledge that I came up with here and go deeper to decide which one of these are definitely going to be wrong. So let's start with A. It says the enzyme is denatured. Well, if this is a normal enzyme reaction right here, the substrate usually shouldn't be affecting, it might change the shape of the enzyme temporarily to allow the substrate to be broken down, but it's not going to change the shape of the enzyme permanently. The very definition of an enzyme is that it's a biological catalyst that can be reused and helps to lower activation energy, etc., etc., etc. So the enzyme is denatured when I increase the substrate concentration. No, that won't happen. But temperature and pH could denature the enzyme, so watch out for that. But in this case, A is not right. Okay, B. Now I'm going into higher level territory. Competitive inhibition is reduced. My knowledge of competitive inhibition reminds me that if I have an enzyme right here, and here's its active site, if this is its active site, normally the substrate is supposed to bind here. But if there's an inhibitor, a molecule that could actually get in the way and block this thing, and it's called competitive inhibition because it actually competes for the same site. It's also the same shape, and so it could bind here, and if it binds here, it'll actually block the substrate, thereby inhibiting it. That is called competitive inhibition. So what happens when substrate concentration is increased? If I increase the number of good guys over here, and these are the good guys, the substrates, can this outcompete the competitive inhibitors, these bad guys around here? And the answer is yes. The way to overcome the effect of these inhibitors is to increase the substrate concentration. So will competitive inhibition be reduced if I increase the substrate concentration? And the answer is yes. So that makes B look like the right answer, and I'm going to circle it halfway. Why? Because I haven't checked these other ones yet. So let's look at C. End product inhibition occurs. End product inhibition occurring is about non-competitive inhibition. I can see D is also about non-competitive inhibition. What are those guys about? Well, if I have an enzyme right here, and this is our active site, there could be another site located somewhere on the enzyme. If you've studied this or this is fresh in your mind, go to BioNinja or your notes or your textbook or whatever to figure out. There could be other molecules that will bind to some place that is not the active site. And when it binds here, it can actually change the shape of the active site. And when it does that, the substrate no longer binds and it just gets rejected. That's called non-competitive inhibition. In a specific version of this, the end product of a metabolic pathway could actually be the non-competitive inhibitor, which will bind here and therefore prevent any more of the end product being formed. There's other videos that exist. I've made one about non-competitive and competitive inhibition, so I can't explain the full thing here, and I've got good diagrams to help out with that. So go review that if this is totally not in your brain right now. So you need to probably go review that. But don't just watch videos blindly. Do these questions, figure out what you don't know, and then go fill the blanks for what you don't know. So end product inhibition and allostery are related, non-competitive inhibition. So, but substrate concentration is only really going for the active site. So these changes here, end product inhibition and the allosteric site being blocked have nothing to do with the substrate concentration actually increasing. That's not going to, the substrate's not actually going to compete for this site. So that both of these are not going to be the correct answers. So hopefully you understood my explanation here. This you really need to know the difference between competitive and non-competitive inhibition. So I'm ready to circle B as the full answer there. Okay.